Hi everyone. In this PowerPoint, I'm going to walk you through the highlights of the BIO paper. This is a study that links genotype with clinical phenotype, so it's a second great example of molecular pathology experiments. Just as a reminder, we discussed molecular pathology for cystic fibrosis in the 331 PowerPoint. So the material in this PowerPoint file will help you complete assignment 4.7, that is due at 11 a.m. on Tuesday, April 7th, and slides 12 through 15 may help you when you are writing part 6 of your research paper, which is proposing new experiments. At the top of this slide, I have a few writing tips of the day. So as you're continuing to work on drafts of your research paper, there's a couple things to look for, avoiding the vague use of it, this, that, they, and which. So you can kind of just do a search in your own drafts and look for those words and then replace them with more specific terms to clarify what it is that you're referring to. A little bit of a grammar reminder about using commas between two independent clauses, like I like apples, comma, and I like pears, right? Because we have a subject and a verb in that second part of that sentence, we need to use a comma to link those two. And just a reminder to try to write simple, relatively short sentences. We're describing complicated ideas, so it's going to be much easier to convey those ideas by using shorter sentences and avoiding lots of phrases within a single sentence. You should use the present verb tense to describe published results. That's information that is known. And then one last reminder, affect is a verb, effect is a noun. So the rain will affect crop growth, but the effect of rain is crop growth. Okay, so those are just a couple things you can look for in your own writing, and when we get to peer editing, I'll be reminding you to look for those things in other people's papers as well. Okay, so we're going to go through this study by Vital. The name of this paper is Complete Androgen Insensitivity Due to Mutations in the Probably Alpha Helical Segments of the DNA Binding Domain in the Human Androgen Receptor. Okay, so what is this paper all about? What did they know before they started? They knew the androgen receptor, which they refer to as AR, is important in mediating testosterone effects for the male phenotype at birth and during puberty. They also knew the androgen receptor binds to the ARE of target genes after it has been bound by its ligand, and they knew that that helped regulate transcription. And on the next slide, I'll show you a figure that helps us understand what that means. They also knew that the AR may regulate localization to the nucleus and dimerization. They knew the protein has 4 domains, which we'll see in the next 2 slides. They also knew that patients with androgen insensitivity had 3 different sort of types of that androgen insensitivity, or 3 degrees. And in the past, diagnosis of AI and prognosis had been aided by studies of GSF, which stands for genital skin fibroblasts. In many patients, up until the point of this paper, mutations had been found in the AR protein in the ligand binding domain. And the authors refer to this sometimes as the androgen binding domain, and that's the same thing. And so in a couple of slides, when we look at a picture of those domains, I'll explain that. In just a few patients, they had seen mutations in the DNA binding domain as well. So that's sort of what they knew about the role of the androgen receptor protein and how it was important in androgen insensitivity. So what was the goal or the hypothesis at the start of the study? Specifically, they wanted to determine the location of mutations in the AR gene for 3 families in which AI, androgen insensitivity, was prevalent. And so they hadn't been quite investigated up until this point. And for 2 of those families, it was known that there was decreased androgen binding by that AR protein. Okay, so this is a picture to show us the pathway in which the androgen receptor works. So if we start at the bottom left, we see testosterone enters into the cell. It's going to get converted, that's the T in the hexagon. It's going to get converted BI5 alpha reductase into DHT, which is dihydrotestosterone. DHT will then bind to AR, which is that yellow puzzle piece looking protein. And once DHT binds to AR will release the HSP90 and HSP70 proteins. So the purple and green proteins will then dissociate from AR, and then AR will be then bound by DHT and then translocate from the cytoplasm into the nucleus. You can also see that it gets additionally phosphorylated. So there's lots of P's that are shown on the AR protein. So those are just phosphorylation sites. And then you can see that AR, that yellow puzzle piece, is going to dimerize. So it's bound to itself. So we see two AR proteins together. And they are binding to the 3' end of a gene that has an ARE, which is an AR element. So that's just a specific sequence of DNA that's a binding site for AR and that whole complex of proteins. So you can see that AR, after it dimerizes, you see the DHT is still bound, and then you can see all these other colored proteins, ARA70, SRC1, CBP, all bind together, and this all is going to help RNA polymerase II bind tightly at the promoter. Therefore, transcription will occur at a high level. So the bottom line here is that binding of AR and its associated proteins promotes transcription of that RNA. And then, of course, after transcription, the RNA will be translated into protein, and that will give that androgenic effect. Okay, so this image shows us how the human AR gene has 8 exons, and below the exons, which are the middle, it shows you the 4 domains of the protein. And these are color-coded, which is kind of nice. So you can see the numbers 1 to 919. That just indicates how many amino acids are in the protein. The first exon encodes that first N-terminal domain. Exons 2 and 3 in yellow encode the DNA binding domain. They don't show us what encodes that little small green domain, but then the majority of the other exons 4 through 8 encode the ligand binding domain. And it's that red domain again that the authors sometimes call the androgen binding domain, because androgen is the ligand that's binding to AR. Right, testosterone is a type of androgen. So again, their question is, in these patients with androgen insensitivity, what is the location of the mutations? They want to get a better understanding of the relationship between the genotype, the specific mutation, and where it is, and the clinical phenotype. Okay, so you can see in those arrows that the white arrow is indicating many mutations have been found in the ligand binding domains, and then the red arrow is showing a few have been found in the DNA binding domain. Okay, so I mentioned that this PowerPoint is going to help you complete the assignment 4.7, so you should make sure to go ahead and get that assignment at this point, because I'm going to kind of walk you through how you can use the figures and tables in this paper to help you complete that assignment. The first question these authors asked was, how well does the AR protein in these patients bind to androgen? So on the left in this table, we have 3 families. You can see that they've shown you what the mutations are, but they didn't actually know that initially. Okay. They have the subject numbers, and then they have what they learned for the Bmax. So the Bmax is a readout of how well the androgen protein in each family member, how well it can bind to androgen. So I've put a little red rectangle around those values, and you're going to want to fill in those values for each of those subjects or patients. And that'll be in the first row in the table for assignment 4.7. Okay, so you can see normal is 15 to 40 femtomoles per milligram of protein. So that at the bottom is our control. And you can see that for all of the family members, the values are lower than that. Some are more significantly lower than others. Okay, their next question, after realizing that the AR protein does not bind androgen normally, is to ask in which domain of the AR protein are the mutations. So they expected to find mutations in that ligand binding domain, right, in the red area. So I've shown you again in the upper right, just to remind you where those domains are. Because they found in the patients in that first table that the AR protein did not bind androgen well, they expected, well, that's probably because there's mutations in that domain. However, when they sequenced exons 4 through 8 of the AR gene in the patients, they did not find any mutations in those areas. So next, they sequenced exons 2 and 3 in the DNA binding domain, and the results of that are shown in figure 1. I'm only going to show you part A of figure 1. So this is comparing normal on the left, the sequence of the exons 2 and 3, or a portion of that sequence. So we have normal on the left, and then patient 12474 on the right. So what we can see is that I've circled in green, there's a missing phenylalanine. There's 3 asterisks, which means that those 3 nucleotides are deleted, and those nucleotides normally would be translated into a phenylalanine. So they would indicate the genotype for individual 12474 as delta Phe 581. So delta means that the phenylalanine at position 581 is deleted. So that's that triangle, is the delta. So that's what you would want to write in your assignment 4.7 table, and you can use parts B and C of figure 1 and the text to complete the remainder of row 2, and to write down the genotypes of the other patients. Okay, so this was a surprise. They found mutations were actually in the DNA binding domain, so now they need to understand a little bit more about what is going on with these different mutations and how are they causing problems. So they wanted to know how much AR protein is translated. They thought this might help explain the differences in androgen binding they saw in table 1. So they did a Western blot analysis, and we can look at the intensity of the band in each lane of the Western blot to figure out how much protein is made, how much of the AR protein is made in each patient. And the control is individual 2080. So we're basically going to compare the 3 other lanes to the band intensity in the 2080 lane. So you can go ahead and do that on your own. You can pause at this point and take a minute to jot that down on your assignment in the 3rd row of the table. A second thing to consider is whether the size of the protein changes in the AR patients. So again, you would just want to compare, does the band seem to be migrating in the same place, which is indicative of size? And if there seems to be any changes in the size of the protein, you can again write that down in your table in your assignment. One thing I'll tell you is that this is very unusual to crop the Western blot the way they have. They're not showing you the entire blot. And in fact, you can see some kind of strange band in between the 2nd and 3rd lanes. So it's very odd to not show more of that blot. And in fact, I think this would not be acceptable in the 21st century in most journals. Okay, but if we take them at their word, we can see that at least some protein is being made in each of these patients, even if you think that it might vary a bit from the control. Okay, so that was about protein levels. Their next question was to ask how much RNA is detected. So how much of the androgen receptor RNA is actually transcribed in the patients. And they basically want to know if the differences they're seeing in protein levels in figure 2 on the Western, is that actually due to a problem with transcription? So they do an RT-PCR assay, and it's a little confusing, this graph. So I'm going to walk you through this. First, you want to find the controls. So that's the 2080 and the 855192. These are the top 2 lines. So these have the highest amounts of the androgen receptor mRNA. So it's the unfilled square and the filled in square. And in the text, they state that the bottom 2 lines, corresponding to 12474 and 75888, that the cells from those patients have much lower amounts of the AR mRNA than patient 15579, which is in the middle, and it's much lower amounts than in the controls. So you can fill in row 4 of the table on your assignment with these qualitative results for RNA levels. And then on your assignment page, you can use the results from figures 2 and 3 that tell you if RNA and protein are made in each of the 3 patients. You can start to answer the questions about whether the mutations in each patient, are they complete nulls, partial nulls, transcriptional or translational nulls? So you want to consider all those questions and answer the question below the table on the 4-7-20 assignment. Okay, so now that the authors have figured out that there is some RNA and protein made, and then they have an idea that androgen binding is not occurring normally, the next thing they want to ask is, can the androgen receptor protein in the patients, does it still bind to DNA normally? So again, I'm showing you that picture of the different domains. And remember, we expect the AR protein normally to bind to DNA. Specifically, remember, it binds those AREs at the 3' end of genes that is shown on slide number 3. So, especially because they found mutations in that yellow DNA binding domain in the patients, you might predict that the AR protein in the patients will not bind to DNA as well. And in fact, that's what they see. So figure 4, in this graph, we are going to consider that y-axis as a readout of how well the androgen receptor protein binds DNA. Note the 2 controls are C and R+, so they have a relatively high % of competition, which means the AR binds DNA well. Okay, so then we can look at the 3 patients, which are shown with the filled in symbols, and you can indicate in row 5 how well the AR protein in each patient binds DNA. And you can state that as a % competition, right? And that's our readout for how well it's binding to DNA. And a note about that x-axis, they are labeling the samples by genotype instead of by patient number in this figure, so that's just a little bit different than in the other figures. Okay, in their last 2 experiments, they were addressing this one question: how well can the AR protein in patients activate transcription of a target gene? And they call this transregulatory competence. So remember, we expect transcription factors are not only going to bind DNA, but they're going to also promote transcription. And we saw that in the pathway for AR. So their readout is how much GH is secreted. So you see that on the y-axis on the left for panel A, and it's on the right part of the axis for panel B. So the GH is growth hormone, and that was the reporter they used to measure transcription. So as you can imagine, the higher the number, that just tells you how effective AR was at promoting transcription of GH. So if we first look at panel A on the top left, we can see that the control, which is the open square, the androgen receptor, is able to elicit quite high numbers or amounts of secreted GH, meaning that it is promoting transcription at high levels. And then one of the patients is shown below that. That's the triangle filled in, delta 614R mutation. And we can see that relative to the control, only very low levels of GH are secreted, meaning that transcription is not occurring well at all. In panel B, they look at one of the additional mutations, the delta 581F. And so let me just walk you through this. We're going to focus on the black bars first, because that's the growth hormone that's secreted. So in this case, it's just a bar graph instead of a line graph, and we can see in the control on the left, the HAR is human androgen receptor. We have a tall black bar, meaning that there's a lot of GH secreted and lots of transcription. We already saw that the R614H, so that is a different one, right? The other one was the deletion of 614. This is the substitution, and it has a similar effect. The black bar is almost at 0, meaning no GH is secreted, and that means it cannot promote transcription. And then the third mutation, delta 581F, also has basically almost a non-detectable black bar, so no GH is secreted, and it also cannot promote transcription. So all 3 of these different patients or different kinds of mutations seem to have a similar effect. and that transcription is not able to be activated. On the left y-axis, it's showing you how much ligand binding can occur. And that's very interesting because in this particular assay, the two mutations, R614H and Delta 581F, seem to be quite similar to the control. So they're able to bind to the androgen, which is MB, but they are not able to then actually promote transcription of those genes that there should be binding to. Okay, so the last thing I want to tell you about in today's PowerPoint are a couple of follow-up experiments and things we could think about if we wanted to better understand the mechanism of gene action or protein. How does the protein work? So in part 6 of your research paper, which will be coming up pretty soon, you're going to need to propose experiments. And one of the types of experiments that you'll be able to choose would be to better understand how these mutant proteins really work. So, what experiments would better help us understand the effects of AR mutations on this pathway? So, if you are lucky enough to find a pathway, and you may use Google and find something like this for your gene of interest, right? You can find the protein encoded by that gene and find a picture of a pathway like this. You can then think about questions like I'm showing you here in blue. So now we know that there are these particular mutations in these patients. You could do yeast-2 hybrid assays to see if the R614H mutant AR protein could still bind to HSP90. We know that it normally binds to the purple HSP90 out in the cytoplasm. It would be interesting to know if the mutant form affects its ability to bind to that protein, or to DHT, or to CBP, or any of the other binding partners that are shown in this pathway. So that would be one kind of experiment you could propose, using a method we've already learned about in modules 1 and 2, but now kind of applying that method to ask a novel question to further understand the protein of interest. In this case, we have this transcription factor, so another question you might want to know is, what are the normal targets, right, of the androgen receptor? Which genes are actually being regulated transcriptionally by AR? So in this generic model, right, we have some double-stranded DNA with an ARE, but it doesn't say what DNA that is. So what are the targets that AR is actually binding? So I'm going to tell you about two new methods we haven't talked about before in order to address this kind of question. Okay, so the first thing we want to know is what kind of interaction are we talking about? Is it an RNA-RNA interaction? Is it RNA-protein? Is it DNA-RNA? DNA-protein? Like what are the macromolecules involved? And in this case, what we're talking about is a DNA-protein interaction. We want to know which DNA is being bound by the androgen receptor protein. So it's a DNA-protein interaction, and one kind of method you can use if you have a candidate DNA in mind is a gel shift assay, which is also sometimes called EMSA, for electrophoretic mobility shift assay. Most people just call it a gel shift. So here's a very simple cartoon, and then I'll explain it further on the next slide. In lane 1, you can see the wells at the top of the agarose gel, and in lane 1, you have just the DNA fragment that has been run in that lane. In lane 2, the DNA fragment has been incubated with the AR protein, and if the protein binds the DNA fragment, you literally see a shift. In this case, they've colored it red, but you see how that is has a higher molecular weight. It's closer to the well, right? It does not have the same size because once that protein binds the DNA fragment, it's a larger molecule. So if you see a shift like that, it tells you that your protein did in fact bind the DNA fragment that you had as a candidate. Okay, so this is a really effective technique, but it only works if you have a candidate DNA in mind. Right. Here is just another example actually from a published paper. So again, just as a reminder, the question that this method answers is, does protein X and particular sequence of DNA, do they interact? And the rationale for this is the idea that protein DNA complexes will migrate through either agarose or acrylamide gels more slowly than DNA by itself. So in this case, they're using a radioactive labeled DNA fragment, like P32 will label DNA. And again, this is the candidate DNA that you think your protein binds. And then you can analyze it on a non-denaturing acrylamide gel, which is the type of gel shown here. And so what we're looking at in lane 1 is just the DNA by itself. That's what the probe is. So PEN01, that's the DNA by itself. So it's a fairly low molecular weight. In the middle lane, they've added in a nuclear extract that has the protein of interest. And you can see a shift because there's a DNA protein complex that has formed. So we see a second band and it's a larger size. And lastly, which I've labeled as number 5 on the left, you can ask if it's a specific interaction by adding an excess of unlabeled DNA fragments. And so they call that a competitor. And what that means is that it basically prevents that interaction from happening. So that's a good control. So in lane 3 on the gel, we no longer see the shift. Okay, so I just wanted to kind of briefly explain what gel shifts are. It's a new method we haven't talked about to study protein DNA interactions. Now I emphasize that one only works, that method, when you have a candidate in mind. But the last method for today, in our last slide, is something called chromatin immunoprecipitation assays. People call these CHIP assays. And this is a really powerful technique because it can be used if you have candidates in mind. That's the targeted approach at the bottom. But you can also use this technique if you don't have any candidates. So just like yeast-2 hybrid can be used to kind of go fishing and ask what binds a protein, you can use a CHIP assay to kind of go fishing to find out what DNA your protein binds to. And I'm not going to go through the details of that method, but depending on the disease that you're studying and writing about in your paper and the protein, that might be something that you would be interested in learning more about. If you ever have questions on methods when you get to that section of the paper, I'll be able to help as needed. All right, hopefully this PowerPoint file and narration will help you with the assignment for 4-7, and hopefully I'll see you all soon.